Okay, so chapter 11 was meiosis. Chapter 12 was all of Mendelian genetics, patterns of inheritance, some exceptions. Chapter 13 is going to take both of those things and kind of fold them together, right? So we're going to talk a bit more explicitly about chromosomes and what they do, right? And kind of what's actually happening, right? We talked about kind of Mendel's observations and the fact that that still works for at least a certain kind of bolus of genes and traits and things we see today. And that we can use his model to compare other traits to and kind of see what maybe is actually happening kind of on the chromosomes or kind of at the DNA level that makes things kind of act not in accordance with Mendel's model. So, chromosomal theory of inheritance. We're going to kind of take this section and brief. It goes through a bunch of stuff that we'll just gloss over. But we're going to use an example of sex-linked inheritance in fruit flies because it's going to help us as we go through this chapter, right? And how this kind of provided evidence for the fact that genes were on chromosomes because it was not always the assumption, right? People knew that there were molecules in your cells that had to be carrying information that caused traits to be inherited. But there was a lot of arguments before we kind of figured out it was the DNA. Like a lot of people thought it was your proteins in your cells that were actually carrying your trait information. So there was kind of this long series of experiments around the turn of the 20th century that kind of got us to the chromosomal theory of inheritance, which states that genes that determine traits reside on the chromosomes, which is kind of something we just already know, but they didn't know. But Walter Sutton in 1902 said, this is what I think. I think your genes reside on your chromosomes. And so we'll look at one example where they actually kind of provided evidence for that there's a relationship between the chromosomes you get in your cells from your parents and the traits that you exhibit once you're born and doing stuff. So Thomas Hunt Morgan did a series of experiments using fruit flies. Drosophila melanogaster. You don't even know Drosophila melanogaster, but that's his name. This is the guy or girl. I don't actually know how to tell male and female flies apart, if I'm honest. But there it is. So Thomas Morgan found these flies, these male flies that have white eyes. So the kind of normal wild type, we call them normal or wild type, like generally speaking, fruit flies you find kind of out doing fruit fly things have red eyes, fair. But he was finding these males that had white eyes, right? And when we're doing these kind of experiments, we call this the mutant type. So there's doesn't mean it's got like X-Men powers or anything. It just means that there's something different about its DNA, right? There's been a mutation in the DNA that's caused it to have white eyes for whatever reason. So he took this mutant male, this wide-eyed male fly, and he crossed it with a red-eyed female, right? It's kind of our, just like Mendel started his, right? You've got your two kind of true breeding individuals, you breed them together. And like we would expect, all our F1 progeny had red eyes. So we concluded that red eyes were the dominant trait. That seems normal, right? That's what happened with pea plants, right? If you crossed a white pea plant and a purple pea plant, you got all purple pea plants. That's not interesting, really. But then he took these F1 progeny, right, and did that second monohybrid cross, right, to get his F2 progeny, but he didn't get a 3-to-1 ratio. Not exactly, right? Because when you're actually doing this, like in a lab, you're not literally breeding like one fly with one fly. You're doing this like hundreds of times with hundreds of flies. And so on average, his ratio was actually slightly more than 3-to-1, but red was still dominant. It was slightly more than 3-to-1 with the more than 3 being red eyes, yeah? But all the individuals with white eyes were male. They were never female. So that was weird. So why were all the white-eyed individuals male? You never got a white-eyed female. Could it be that white-eyed females are non-viable? Maybe there's something, right? Maybe it's a yellow mouse situation, right? Maybe you can't have a white-eyed female. Maybe they don't develop properly. It's like, let's do some more experiments. So, to kind of tease this part of it.
How do you get fruit flies to breed?
It's harder to not have fruit flies breed, honestly, in the big scheme of flies. You just kind of put them in the same place. Yeah. Yeah, you just keep them in little tubes and you just put like these two in a tube and it handles itself. Yeah. I mean, there's other bugs that are more difficult. Flies are not difficult. Yeah, they're all about it.
I thought flies die in like a couple weeks.
Yeah. I mean, you probably only get like one round out of each one, but you just kind of, I mean, you just keep making more of them. Yeah. Dealing with fruit fly cultures is horrible. Side note, I did it like once during my master's and I did not like it. I was feeding them to plants though, which was weird. Anyway, all right, so he took his female F1, right? This heterozygous, red-eyed female, right? Dominant red allele, and crossed it with a white-eyed male. And now, when he did this, also, what does this look like? I don't know. It's on the slide. What's he doing? He's doing a Testcross. He's doing a Testcross, right? He's got this kind of unknown individual. He crosses it with a true breeding, recessive individual, trying to figure out what's going on. And then he gets really weird stuff, right? Because now, because this male and female thing has kind of come into play, he basically ends up with a 1:1:1:1 ratio, which has never happened to us. What the heck? So white eyes and red eyes of both sexes. So he's got a red-eyed female, a white-eyed female, and a red-eyed male, and a white-eyed male. This is breaking all of our rules. This is really obnoxious. So he determined that the nature of the chromosomes in males and females, because at this point, we didn't really get what DNA was doing, but we knew, right? We could see DNA, we could see chromosomes. We had a concept of X and Y chromosomes. And so he's like, there's some relationship between the sex chromosomes and the eye color, which seems weird. But there's some kind of interaction, right, going on. So, turns out that this is a sex-linked gene. What does that mean? So sex in fruit flies, right, biological sex is determined by the number of X chromosomes, same as us. If you get 2 X chromosomes, you are biologically female. If you only get 1 X chromosome, you are biologically male. And if you only get 1 X chromosome, it is generally paired with a Y chromosome, right? So we define these, right? These X and Y chromosomes that have to do with kind of the determination of biological sex. We call them sex chromosomes. Easy enough, right? And so we've kind of had them on the squares and not been talking about them, but these chromosomes are all X chromosomes, right? So the female has 2 of them. Male only has 1, right? He's got 1 X and 1 Y. Right? Y over there. Okay, so what? Morgan determined that the gene causing white eyes must reside on the X chromosome, right? And is absent from the Y chromosome, right? This is where your sex chromosomes are kind of different from all your other chromosome pairs, right? They don't match. So the gene for eye color is on the X chromosome. And so traits, we've got a name for this, traits determined by genes on the X chromosomes are sex-linked, or sometimes we just say X-linked, right? And so this was kind of the first real kind of clear evidence for the chromosomal theory of inheritance, right? That, right, obviously these chromosomes were carrying the traits he was seeing in the flies, right? It wasn't other weird proteins in the cell that were passing this information. It was the DNA, right? So let's kind of build this up a little bit, right? So we've got chromosomal basis for inheritance. We have sex-linked traits. Let's ramp it up a little bit. So we've got sex chromosomes. We've discovered them. We see them. And then we have sex determination, okay? So there's a relationship between sex chromosomes, what you get, what mix of different chromosomes you get, and your biological sex. And then we'll talk a bit about dosage compensation, should we get that far. We might. All right. So sex determination in animals is based on the presence, absence, or number of various sex chromosomes. And it varies from like group to group, right? So us and the flies, right? If you inherit 2 X chromosomes, your biologic is female. If you inherit 1 X chromosome, it pairs with a Y chromosome and you're biologically male. If you're a bird and you inherit a Z and a W chromosome, you're female. You get 2 Zs, you're male. Grasshoppers, similar to us, if you inherit 2 X chromosomes, you're female. Males have an unpaired X chromosome. So they inherit an X and then nothing. Honeybees, if you're diploid, you're female, and if you're haploid, you're male. The males are all just haploid clones. There's no actual fertilization in their production. In spiders, some spiders have hundreds of sex chromosomes, and it's just kind of like a tipping point. If you get enough of whatever the spider's sex chromosome is, you are female. If you don't get enough, then you are male. I think there's one group that's like 140 something sex chromosomes. It's crazy. So everything is different. It can be quite complicated. It's relatively straightforward-ish in mammals and the fruit flies. But the presence, absence, or number of them or type of them will determine biological sex. So, like I said, in us, we have 22 chromosomes that pair perfectly, right? Are not involved in sex determination and that are appropriate, lined up, nice, can cross over, tidy little paired chromosomes. Those are called autosomes. That last pair number 23, are the sex chromosomes, right? 2 X's in biological females, an X and a Y in biological males. The Ys, right, kind of carry very specific instructions. So they're highly condensed. They don't really actually have a whole lot of genes on them, which results in recessive genes on the X being expressed in males because there's no counterpart, right? I can have a recessive gene on one X and a dominant gene on the other X, and I'll express the dominant one. But because a male only has one X, it will express recessive traits because there's no counterpart. Another thing that goes along with this Y chromosome situation is that the embryonic default is female. The Y chromosome actually carries a specific gene, the SRY gene, that switches embryonic development to code for male development. All right, so what was happening in the flies then is that there was this, he was not wrong, right? The red allele was dominant, the white allele was recessive. But for a female to have white eyes, it needed to inherit 2 white alleles. The male only had to inherit 1, right? And then that recessive white trait would be expressed. Right? And there are several things that work like this. We'll give a couple examples of genetic, and they're not all diseases, obviously, but there are some genetic diseases or disorders that will affect biological males more than biological females because of that problem, because you can inherit a recessive trait on your X chromosome, and if you only have 1 X chromosome, then you'll express whatever the thing is, right? So a couple of these recessive traits that are kind of like the classic examples, red-green color blindness. It's kind of a classic example. It's why you have more males that are red-green colorblind, because it's a recessive trait, but it's carried on the X chromosome. And then hemophilia, which works the same way. And hemophilia is actually kind of one of these epistasis situations where hemophilia is a change in a gene that codes for one little protein, right? One little enzyme, but that enzyme is important in a cascade of proteins that are involved in blood clots, right? So you basically, right, it's that biochemical pathway problem. We break one step and the whole process breaks down. Your blood not being able to clot is really bad if you were not aware. It's important that your blood clots, right? But we run into this red eye, white eye, similar problem. A heterozygous female is an asymptomatic carrier, right? I could be carrying an allele for hemophilia and have no idea, right? Because I'll just express the dominant allele that allows my blood to clot normally, One of the kind of classic, and I think sort of fun examples is Queen Victoria. She was kind of fun and weird. But Queen Victoria was a heterozygous carrier of hemophilia. So she was fine. She was not unwell. She had roughly 1 million children, right? And due to independent assortment, a few of them picked up that recessive allele, not all of them, right? On this chart, females are circles and males are squares. So any female that inherited it, right, got to be heterozygous, got to be fine, but could pass it on, right? Any male that inherited it just had it and definitely passed it on, right? Because it only had one pass on, right? So Alice and Beatrice passed it kind of on to their children. Edward and Victoria here got kind of lucky with no hemophilia getting passed on. And so the upshot of this particular situation is that Queen Victoria passed her hemophilic alleles onto the Prussian royal house, the Russian royal house, and the Spanish royal house, and basically killed off the lot of them inadvertently, right? Because all of these, you see all these males, most people, males, most anybody who has hemophilia does not survive childhood. Like, how many times did you scrape your knee as a kid? If the option is you bleed to death from scraping your knee, right? It is really hard to get past being a kid. So any males who got this allele, basically, other than Leopold, must have just been very careful. Like, maybe they kept him like wrapped in blankets or something, right? And then just by chance, turns out the British royal house of Queen Victoria's line made out scot-free. They're fine. Well, at least as far as hemophilia is concerned, they've got other issues. Yeah, so hemophilia is a sex-linked genetic disorder. All right, just a few minutes. Yeah, there's Queen Victoria, we know. So last little piece of this story is dosage compensation, right? Because now I know, right? A biological male will have one X chromosome and just express everything on it because he's only got the one. I have 2 X chromosomes. So do I just express everything on both of them? That seems wrong. I don't produce twice the amount of all of the things that are being coded for. And so in females, in each cell, right, in all of your cells, one of the X chromosomes is actually just turned completely off, right? It's turned off, it's stuck to the side of the nucleus, and it doesn't do anything. It's called a bar body. So you express all of the alleles from one of your X chromosomes, and the other one is just taken out of rotation. And this inactivation of one of these female X chromosomes is called dosage compensation, right? It's compensating for you having kind of a double dose of X chromosomes relative to the rest of the population. This can result in some kind of fun things, I think, like calico cats, right? Because depending on, in a given organism, the alleles that are carried on that X chromosome and which of the X chromosomes gets turned off, what's happening in these, your kind of classic black, brown, and white calico cats is that you have a female who's heterozygous for a trait, and they exhibit a mosaic trait depending on which allele is inactivated in every region of their body, right? So wherever this cat is brown, the allele for black fur was deactivated as the bar body. Wherever the cat is black, the orange fur allele X chromosome was deactivated. That's where calico cats kind of, that's how you get a calico cat, right? There's another gene that comes into play that makes it, more complicated, but there's a second gene that causes it to be kind of patchy across the body. White fur is just where there's no pigment for other reasons. But anyway, you get this genetic mosaic situation depending on which X chromosome is deactivated. Okay, I think that gets us to yes, because we can't talk about something without immediately following it with exceptions. We'll start Monday with exceptions to chromosome theory. So, which I think, do you look at these slides? It may be one of the questions on the quiz now that I think about it. I think I did go as far as I meant to. but I can just always go in and fix that. All right.